Yesterday you did some reading about meiosis, which is how cells are going to form gametes in sexual reproduction. So what I want to do today is I want to try to compare mitosis and meiosis so that you really understand the difference between the two of them. Meiosis is twice as long as mitosis because the cells have to divide twice. But where it can get confusing sometimes is what's actually happening with some of those chromosomes. So that's why I want to go through and try to do a comparison for you so that hopefully it will make some more sense. Both mitosis and meiosis start with a diploid cell. Now, what's a diploid cell? A diploid cell means that you have homologous pairs of chromosomes. So what that means is you got a set of chromosomes from your mom and you got a set of chromosomes from your dad. And so you have chromosomes for eye color from both parents. You have chromosomes for hair color from both parents. You have chromosomes for your height. You have chromosomes for lots of different things from both parents. And so they're not identical to each other. The two sides are not identical, or rather the two sets are not identical, but you do have both of them. Now, all of those genes are not expressed, and we'll talk a lot more about that in the next chapter, but you do have all of those chromosomes in every single cell. And so the chromosomes are going to kind of pair themselves up so that similar traits that are coded for are going to be on the homologous chromosomes. So the diploid cell has homologous chromosomes, which means, again, you have one from each parent. Now, in meiosis, what happens is that in prophase, remember that the P stands for prepare, what happens is that you've got little chunks of these chromosomes that kind of swap places with each other. Obviously, you notice that you don't look exactly like your mom. You don't look exactly like your dad. Your dad doesn't look exactly like your grandpa. You've got traits from both of them. And it is this crossing over stage that allows organisms to be able to pass on certain traits, but they're not making exact duplicates of themselves. So when these chromosomes start crossing over, that allows for all the variety we see in all of the different creatures that are going to reproduce sexually. So that crossing over happens. Then in metaphase, remember metaphase means that M stands for middle. And here the homologous pairs are going to line up with each other. Okay, so these two are similar. They may code for similar things, but they're not identical to each other. And so all of these pairs are all lined up as pairs. Then in anaphase, the two pairs are separated from each other. And then when you get down to telophase and cytokinesis, remember anaphase means they're going to pull away, they're going to pull apart. Then in telophase, what we end up with is haploid cells. So I now have half the number of chromosomes that I did before. You notice in my example here, this initial cell had 6 chromosomes. Well, now each cell only has 3. Okay, so I have haploid cells. They have half the number. That's what that H, you can remember the difference between haploid and diploid because haploid stands for half. The H stands for half. So now I have 2 haploid cells, and they have non-identical sister chromatids in them. So everything in them is not matching up like it did before. So if I were going to use my ends and my 2 ends, like you learned that's how we code for diploid and haploid cells. When I start up here, I have a 2-end cell because it's got double of everything. Okay, so in prophase, I'm still at 2N. In metaphase, I'm still at 2N. In anaphase, I'm still at 2N. And then in telophase, that's when everything has pulled apart and said, ta-da! And so then half the cell or half the chromosomes are going into each new cell, and each cell is now just N because it doesn't have as many chromosomes as it had to start with. Okay? So meiosis 1 is way different than what we talked about in mitosis. When we talked about mitosis, in prophase, there was no crossing over. There was just simply a duplication of the chromosomes, but no crossing over happened. And so every daughter cell is going to have exactly the same chromosomes that the mother cell had. Okay, so in metaphase, in place of lining up in homologous pairs, in metaphase, they're all lining up separate from each other, okay? So the diploid cell is in 2N. So here I'm in 2N. Here I'm still in 2N. Now in anaphase, when they start to pull apart from each other, they're detaching themselves at the centromeres. And so for a very brief moment in time, I actually have double the number that I had before. So for a very brief moment here, I'm actually at 4N. And then once telophase and cytokinesis occurs, this is going to be in one cell, this is going to be in the other cell, and I'm going to be down to 2N, ta-da, with 2 identical diploid cells to the one that I started with. Now, meiosis 2 is similar to mitosis in that there's not going to be any crossing over again here, and every single chromosome is going to line up. Now, up here, they lined up in pairs. Here, they're not lining up in pairs anymore. They're all lining up separate, and you can see that when the division happens, they look much different than it did when the division happened here. Okay, so again, these chromosomes are pulling apart at the chromatids, and so here, since I've started with haploid cells, I'm still at N here. My metaphase is at N. My anaphase, for a very brief moment in time before this cell completely separates, I'm back to a 2N, but then once they pull apart, I'm going to be back to an N. So ta-da! I now end up with 4 different haploid cells. Okay? So differences here are that in meiosis, you've got your homologous pairs lining up, whereas in mitosis and meiosis 2, the chromosomes line up singly and not in pairs. Okay? You'll also notice that in all 3 of these stages, there is a very brief moment, well, not in all 3 of them, only 2 of them here, where the number of chromosomes doubles until the cell actually separates. But here I'm going to end up with identical cells to what I started with, and here I'm going to end up with 4 cells very different than what I started with. Here with meiosis, I end up with 4 cells. With mitosis, I end up with 2. Now, your book seemed to indicate that in a female, that the one of the cells that's produced here took all the cytoplasm, and that part's true, and then the other one is left with what's called the polar body. It doesn't have much cytoplasm, and there's not a whole lot there. Your book indicated that the polar body did not go through meiosis 2, and I'm not familiar with that. Everything else that I have seen says that the polar bodies also do go through meiosis 2, but then again at the end, you're only going to have 1 cell in a female, 1 egg, that's actually going to be functional, and you're going to have 3 polar bodies that are not functional because that 1 egg kind of robbed all the cytoplasm from the rest of them. Okay? With males, however, those 4 different haploid cells are all going to be sperm, and they're all functional. Okay, so for the end of the process here, for females, you have 1 egg and 3 polar bodies that are not functional, but for males, you're going to have 4 sperm, and they are all functional. Okay, so hopefully this diagram helps you make some sense out of the difference between mitosis and meiosis. Mitosis and meiosis 2 are very, very similar to each other as far as how the chromosomes line up and how they divide and what ends up happening, because here in mitosis, I started with 2N cells and I ended up with 2N cells. In meiosis 2, I started with Ns and I ended up with Ns. But in meiosis 1, this is the place where that N number gets divided in half by the end of the process. Okay, so meiosis 1, then meiosis 2, 4 haploid cells, and then mitosis ends up with 2 identical diploid cells.